Welcome back to the Deep Dive. Today we're tackling a system that is, it's both mechanically brilliant and, frankly, often ignored until it breaks.
It really is.
We're talking about the peripheral vestibular system. This is that secret sense, that lightning-fast system that keeps you upright and almost magically keeps your vision stable.
Even when you're running, it's
Mm.
locking your gaze.
Exactly. It's the hidden physics of balance, and it's all happening deep, deep inside your inner ear. We've got a fantastic source today, and our mission really is to be meticulous. We're going to go through this thing in excruciating detail, piece by piece.
Absolutely. And, for anyone listening who really wants to understand the nuts and bolts, this is it. We're going to build this up from the ground floor. We're talking about the anatomy, the fluids, these tiny hair cells, and then the brainstem pathways that make sense of it all.
So we're going to treat our source like a blueprint. We'll start with the structure, the casing that protects it all, and then drill down into the microscopic world of the actual sensors.
Sounds perfect. And to really appreciate the inner ear, we have to just quickly place it in context. The old three-part division of the ear is probably the best place to start.
Right. So the external ear, we can move past that pretty quickly. It's the pinna, the part you see, and the ear canal, all leading up to the eardrum.
The tympanic membrane, yeah. Its job is basically to funnel sound in.
Then we get to the middle ear, and it's a bit more complex. It's this small air-filled space.
And it's bounded by the eardrum on one side and then this complex bony wall on the other side with little windows in it, the oval and round windows.
And inside the space, you've got the famous acicula chain, the tiny bones.
Malleus, incantus, and stapes. And this is where we see the first piece of just incredible engineering, even though it's for hearing, not balance.
You're talking about its function as a transformer mechanism. Why? Why is that so important?
It's absolutely essential because it has to solve a huge physics problem. It's what scientists call an impedance mismatch.
Impedance, so that's just resistance to energy flow.
Exactly. Think about it. Sound is just pressure waves moving easily through air, which has, very low impedance. But it needs to transfer that energy into the fluid of the inner ear.
Which is like trying to shout underwater. Most of the sound just bounces off the surface.
Precisely. The fluid is high impedance. It resists the energy. So without this transformer, which uses a large eardrum to focus all that force onto the tiny little oval window via the bones, more than 99% of the sound energy would just reflect away.
So it's a mechanical amplifier, basically.
That's a perfect way to put it. It boosts the pressure to make the fluid move.
Okay, so that context is set. Now let's go deeper to the inner ear itself. This is the home of the vestibular system, and it is incredibly well protected.
It's housed within the petromastoid part of the temporal bone, which is, it's the densest bone in the entire skull.
A natural stronghold.
It really is. This bony casing, this structure, is what we call the bony labyrinth.
And even within this one structure, there's a clear division of labor, right?
A very clear one. The bony labyrinth contains two distinct functional parts. One is the cochlea, which is coiled like a snail shell. That's for hearing. And the other part is our focus today, the peripheral vestibular system. Its whole job is detecting head movement static, linear, and angular.
Okay, so if we could, crack open this bony labyrinth, what would we find inside?
Fluid. The first of two very important, very different fluids. This one is called perilymph.
And our source makes a really interesting point here. This isn't just some isolated fluid. It's connected to the brain.
It is. The perilymph actually communicates directly with the cerebrospinal fluid, the CSF, that bathes the brain and spinal cord. So its pressure and composition are linked to the whole central nervous system.
But that's not the whole story, because there's another labyrinth inside the first one.
Yes. This is the critical part. Nestled inside the perilymph-filled bony labyrinth, kind of floating and supported by connective tissue, is the membranous labyrinth.
And this is where things get really specialized. It's described as an anatomically and biochemically distinct closed system.
It's completely sealed off, which is so important because that allows it to maintain its own unique internal fluid.
Which is our second fluid, endolymph.
Endolymph. And its chemistry is, frankly, bizarre. Unlike almost every other extracellular fluid in the body, which is rich in sodium, endolymph is incredibly rich in potassium.
That's a chemical profile you normally see inside a cell, not outside it.
Exactly. And that biochemical oddity is the secret to the whole system. That high concentration of potassium creates a massive electrical battery that powers the sensory hair cells. We'll definitely come back to that.
So let's just recap the foundation here. We have a super protective bony cage filled with one fluid, the perilymph. And inside that, we have this delicate sealed bag, the membranous labyrinth, filled with a second electrically potent fluid, the endolymph.
That's the architecture. And that membranous labyrinth is made of 5 interconnected segments, each designed to detect a different kind of motion.
Okay, so let's unpack those 5 segments. We need to know what they are and, what specific movements each one is built to detect.
Right, so the 5 segments are really 2 types of receptors. First, you have what are called the otolith organs. These are the saccule and the utricle.
And what's their job?
They detect 2 things. Static position. So it's the tilt of your head relative to gravity and linear head movement.
Linear. So moving in a straight line, like accelerating in a car or going up in an elevator.
Exactly that. Acceleration or deceleration, horizontally or vertically. That's the otolith's job.
Okay, so that's 2 of the 5 segments. What are the other 3?
The other 3 are the semicircular canals. You have a superior, a posterior, and a lateral one in each ear. And their job is singular. They detect head rotation, angular movement.
Turning your head, nodding, tilting side to side.
Any kind of rotation. And what's so brilliant about them is their physical arrangement.
They're set up in orthogonal planes.
Yes, approximately orthogonal to each other. You can think of them like the 3 axes on a graph, an X, a Y, and a Z axis.
Yeah. Which means no matter how I rotate my head, some combination of those canals will be stimulated. It gives you a full 3-dimensional sense of rotation.
It's a complete 3D motion detection system.
Yeah.
And they don't work alone. They work in functional pairs.
Right. It's not just the 3 on the left working together. It's about how the left and right ears coordinate.
Precisely. They're organized into 3 pairs. The 2 lateral or horizontal canals work together. When you shake your head no, one is excited while the other is inhibited.
A push-pull system.
A perfect push-pull system. Then you have the cross-body pairings for vertical movements. The superior canal on one side is paired with the posterior canal on the other side.
Oh, wow. So it's diagonal.
It's a diagonal pairing, and vice versa.
Yeah.
The posterior on one side pairs with the superior on the other. This setup guarantees that any rotation in any plane gives the brain this perfectly clear, contrasted signal. Excitation on one side, inhibition on the other.
It's such an elegant design. Okay, let's go back to the linear detectors. The saccule and the utricle, the sensing happens in specific little patches, right? The macula.
That's right. The sensory cells are clustered in these 2 regions called the maculae. And again, their orientation is everything.
So the saccule macula, what's its orientation?
The saccule's macula is oriented vertically, so it principally detects linear acceleration and head tilt in the vertical plane.
That's the elevator sensor.
It's the elevator sensor, and it's also what feels it when you nod your head, yes.
And the utricle then must be horizontal.
Correct. Its macula is in the horizontal plane. It's what detects you accelerating forward in a car or tilting your head to your shoulder. And crucially, the 2 maculae are set at 90 degrees to each other.
Again, covering all the axes, but this time for linear motion.
Total coverage.
Okay, now this is where the source gets really interesting for me. The actual mechanics of how the maculae work, it involves this thing called the otoconal membrane.
Yes.
Yeah.
It's a masterpiece of biological physics.
Yeah.
The little hairs from the sensory cells, the cilia, project up into this sheet. It's a fibrocalcareous sheet.
Fibrocalcareous, so fibrous and chalky.
Exactly. And the most important detail, the thing the entire mechanism depends on, is that this membrane has a specific gravity that is much, much greater than the endolymph fluid around it. It's heavy.
Why is it so heavy?
Because it's loaded with millions of tiny, dense crystals of calcium carbonate. They're called otoconia, or sometimes just ear rocks.
Ear rocks, I like that. So you have this heavy crystal-laden blanket sitting on top of the sensors.
A perfect analogy. And because it's heavy, it has inertia. So when you tilt your head, gravity pulls on this dense membrane, causing it to shift. Or when you accelerate forward, the membrane lags behind for a split second due to its inertia.
And that movement, that slight lag or shift,
Creates a direct shearing force. Yeah. The heavy blanket slides just a tiny bit across the tops of the hair cells, bending the cilia. And that bend is the trigger for the whole neural signal.
that is just incredible. So it's using density and inertia to detect both gravity, which is a constant force, and acceleration, which is a transient one.
2 different physical phenomena detected by the same simple, elegant mechanical system.
And the source mentions a feature called the striola that helps organize this.
The striola is a little depression or a line running through the macula. And it's basically a dividing line for the polarity of the hair cells. On one side of the striola, the cells are organized to be excited by movement in one direction, And on the other side, they're excited by movement in the opposite direction.
So it's not just detecting movement, it's detecting the precise vector of that movement across a surface.
It gives it much finer spatial resolution. It's a brilliant bit of wiring.
Okay, let's pivot now to the rotational detectors, the semicircular canals. The sensory part is in a specific spot called the ampulla.
Right. Each canal has this little bulge at one end, the ampulla. And inside, there's a crest of tissue where the hair cells sit. And their cilia, their little hairs, stick up into a very different kind of structure.
Not a heavy rocky membrane this time.
No, this time it's a buoyant gelatinous mass called the cupula. And unlike the otoconal membrane, the cupula has the exact same specific gravity as the endolymph around it. It just floats there.
So it's not sensitive to gravity or linear acceleration at all?
Not at all. It's neutrally buoyant. It acts like a little swinging door or a sail inside the canal.
So when you rotate your head, the canal and the cupula move, but the fluid inside, the endolymph, lags behind because of its own inertia.
Precisely. The fluid effectively flows in the opposite direction of the head turn. And this fluid drag pushes on the cupula, deflecting it.
And that deflection bends the cilia of the hair cells embedded within it.
And that's the signal for rotation. So you have two completely different mechanical systems tailored perfectly for two different kinds of motion. The otoliths use a heavy, dense membrane for linear forces, and the canals use a light, buoyant sail for rotational forces.
This raises the big question then, the one that we absolutely have to dig into. How does a mechanical bend, a shearing force, actually become an electrical signal the brain can read? It all comes down to the hair cells themselves.
Right, and the sheer number of them is staggering. Our source says there are about 63,000 hair cells in each ear's vestibular organ.
63,000 individual sensors. And most of them, about 40,000, are in the maculae for linear detection, with the other 23,000 in the canals for rotation.
And the entire mechanism is happening on the very top surface of these tiny cells. Each one has a bundle of about 50 to 100 little non-moving cilia, the stereocilia.
And the key word the source uses is asymmetrically arranged. They're not just a random patch of hairs.
Not at all. They are meticulously organized in a stepwise fashion, like a tiny pipe organ getting taller and taller towards one side. And at the very end of that row is the single longest, most important cilium, the kinocilium.
The kinocilium, that sets the direction for the whole cell, doesn't it?
It establishes the cell's polarity. It's the this way is on arrow for the sensor.
And there are little tiny physical connections between these cilia, the tip links.
The tip links are the magic ingredient. They're these tiny little protein filaments that connect the tip of a shorter cilium to the side of the taller one next to it. And at the end of that tip link is a little ion channel, like a tiny trapdoor.
So when the bundle of cilia gets bent,
the tension on those tip links changes. It's a direct mechanical pull.
Which brings us to the absolute fundamental rule of this entire system, the polarity rule.
This is the binary code. It's beautifully simple. If the bundle of cilia is bent towards the kinocilium, the tallest one,
the tip links get stretched.
You get stretched, and that tension mechanically pulls open those little ion channels. That's the on signal. It causes depolarization, and the nerve firing rate shoots way up above its resting baseline.
Excitation.
Excitation. Now, if the bundle is bent in the opposite direction, away from the kinocilium,
the tip links go slack.
They go slack, the tension is released, and the channels slam shut. This causes hyperpolarization. The nerve's firing rate drops way below its resting baseline. Inhibition. So the system isn't just an on-off switch. It's constantly reporting the precise direction and magnitude of movement based on how much it deviates from that baseline firing rate.
So the mechanical pull opens a pore. Let's circle back to the endolymph. How does that create the electrical current?
It's that huge electrical and chemical gradient we mentioned. Remember, the endolymph bathing the cilia is packed with potassium and has a positive electrical charge, about plus 80 millivolts.
And the inside of the hair cell is negative, like most cells, around negative 60 millivolts.
So you have this enormous 140 millivolt driving force sitting across the membrane at the tip of the cilia. The moment that channel is pulled open, positively charged potassium ions from the endolymph just flood into the cell.
It's a massive instantaneous rush of current.
A huge transduction current. That depolarizes the cell, which triggers neurotransmitter release at its base, and that tells the afferent nerve to fire like crazy.
And even when you're perfectly still, there's always some activity.
Absolutely. The system is never truly off. The afferent nerves maintain a continuous baseline discharge, a steady hum of about 70 to 100 spikes per second. This baseline is perfectly symmetrical between the two ears when you're at rest. Every signal the brain gets is just a change in increase or a decrease relative to that constant steady hum.
Now, we have to get this next part exactly right because the source is very specific and it's easy to get confused. The directional polarity in the semicircular canals.
Agreed. Let's be meticulous here. The on direction is different depending on which canal we're talking about. For the lateral or horizontal semicircular canals, the cells are arranged so that endolymph flow towards the utricle causes depolarization. That's the excitatory direction.
Okay, flow towards the utricle is on for the horizontal canals.
Right, and consequently, flow away from the utricle is inhibitory. It causes hyperpolarization.
But for the other two canals, the vertical ones, it's the opposite.
It is the complete reverse. For the superior and posterior semicircular canals, the kinocilia are oriented on the other side. So for them, flow towards the utricle is actually inhibitory. It's flow away from the utricle that is excitatory.
Wow. Oh, the different arrangement.
It's all about making the push-pull pairing work. This reversal ensures that when you nod your head forward, for example, the superior canal on one side gets excited, while its partner, the posterior canal on the other side, gets inhibited. The brain receives a clean, unambiguous, symmetrical push-pull signal for every possible rotation.
It's an incredibly complex wiring diagram, but it all serves that one goal of perfect contrast. So, we have the signal, it's been generated. Now what? Where do these, 15,000 nerve fibers from each ear? Where does all that data go?
It streams directly into the brainstem to a central processing hub called the vestibular nuclear complex.
Okay, the central hub. And it's located right on the floor of the fourth ventricle, a pretty strategic spot.
A very strategic spot.
Yeah.
It's the main gateway between the peripheral sensors and all the motor control centers of the brain and spinal cord. And this complex isn't just one big blob of neurons. It's divided into 4 distinct regions, 4 nuclei.
And the genius of that, I assume, is that they each have a different job.
A very specific job. The inputs from the different parts of the labyrinth, the canals, the utricle, the saccule, are not just dumped in randomly. They are channeled to specific nuclei, which then project to specific motor systems. It's highly, highly organized.
Let's break them down. Let's start with the one that's mostly for the eyes, the superior vestibular nucleus.
Becquerel's nucleus, yeah. This one gets its input almost exclusively from the semicircular canals, the rotation detectors. So its primary job is managing eye movements during head rotation.
And it does that by sending its output through a specific pathway, the MLF.
The medial longitudinal fasciculus.
Okay.
You can think of it as the superhighway for eye movement control. It's a bundle of heavily myelinated superfast axons connecting the vestibular nuclei directly to the cranial nerve nuclei that control the eye muscles.
So the superior nucleus is the main relay center for the vestibulo-ocular reflex, the VOR.
It's the VOR command center. It ensures your eyes move at the exact same speed, but in the opposite direction as your head turns.
Okay, nucleus number 2. The medial vestibular nucleus. This one seems to have a broader role.
It does. It gets input from the utricle, the horizontal linear detector, in addition to the semicircular canals. So it's starting to integrate more information, and its outputs are more widespread. It sends signals down to the neck and upper back muscles via the medial vestibulospinal tract.
So this one is controlling head and neck movements, keeping your head stable on your shoulders.
Exactly. But it also has these really important commissural connections.
Meaning it talks to its counterpart on the other side of the brainstem.
A lot. This crosstalk is critical for compensation. If one ear is damaged, it's the medial nucleus that helps the brain recalibrate and learn to function with the altered input. It's the recovery center.
Fascinating. Now for the big one for posture, the lateral vestibular nucleus, or Dater's nucleus.
This is the master of postural control for the entire body. What's unique here is its inputs. It gets information from the labyrinth, of course, but it also gets a massive amount of input directly from the cerebellum.
The cerebellum, the brain's main coordinator of movement, so it's getting error correction signals.
Exactly. The cerebellum is constantly comparing the intended movement with the actual sensory feedback and telling the lateral nucleus how to adjust. And its output is just as sophisticated. It projects down the lateral vestibulospinal tract.
And the source says this projection is somatotopically arranged. What does that mean?
It means it has a body map. Specific neurons in the lateral nucleus control specific muscle groups in your legs, your arms, your torso. It's not just a general tense-up signal. It's an incredibly precise, targeted command to specific extensor muscles to keep you from falling over. It is the most important center for vestibulospinal reflexes.
And the last one, the inferior or descending vestibular nucleus.
This one is more of an integrator. It gets input from both the labyrinth and the cerebellum, like the lateral nucleus, but it sends most of its output back to the cerebellum and to another area called the reticular formation. It's thought to be involved in integrating vestibular information with other sensory systems and with autonomic functions.
So if we put it all together, it's this beautiful division of labor. Superior for the eyes, medial for the neck and recovery, lateral for the whole body stance, and inferior for integrating it all.
It's an incredibly efficient neural design. And all of this complex hardware and wiring is constantly working in the background to achieve two main goals: gaze stabilization and postural control.
And it does this through three main reflexes that are just running automatically all the time.
All the time. The first one is the reflex that saves you from a sudden slip or trip, the vestibulospinal reflex, or VSR.
This one has to be lightning fast.
Faster than thought, it's for the rapid correction of posture. If you suddenly start to fall to your left, it causes the extensor muscles in your left leg to contract instantly to push you back upright.
So it extends the limbs on the side you're falling toward.
It extends the ipsilateral, or same-side limbs, and contracts the contralateral, or opposite-side limbs, to stabilize your core. And it's mediated primarily through that powerful lateral vestibulospinal tract coming from the lateral nucleus.
Okay, then there's the writing reflex. This one sounds more about orientation.
It is. Its job is to keep your head level with the horizon, regardless of what your body is doing. If you're leaning over to pick something up, this reflex adjusts your neck muscles to keep your head, and therefore your visual world, stable and horizontal.
And that one is run by the medial vestibulospinal tract.
Primarily, yes.
And then the one that gets tested in the clinic all the time,
Right.
the vestibulo-ocular reflex, the VOR.
The VOR, its whole purpose is image stabilization.
Ah.
It's what allows you to, say, read a sign while you're walking or jogging. Its performance is incredible.
The gain is meant to be 1-to-1, right?
Ideally, a gain of 1.0. For every degree your head moves one way, your eyes move a perfect degree the other way.
Let's trace that signal path just to see the speed. Let's say I turn my head very quickly to the right.
Okay. That head turn causes the endolymph in your horizontal canals to lag, pushing on the cupullae. In your right horizontal canal, the flow is excitatory. The firing rate from that nerve might jump from 80 spikes per second up to, say, 150.
And on the left side, it's the opposite. The flow is inhibitory.
Correct. The firing rate on the left might plummet down to 30 spikes per second. So the brain gets this massive, unambiguous signal. 150 on the right, 30 on the left. The only possible interpretation is a fast head turn to the right.
And it immediately needs to move the eyes to the left to compensate.
Instantly. The excitatory signal from the right vestibular nucleus shoots across that MLF superhighway. It activates the nucleus for the lateral rectus muscle of the left eye, pulling it left, and it activates the nucleus for the medial rectus of the right eye, also pulling it left.
And the inhibitory signal from the left side helps relax the muscles that would pull the eyes to the right.
Exactly. It's a simultaneous push and pull, and it happens in milliseconds. It's the fastest reflex arc in the human body.
And understanding this concept, this absolute reliance on signal symmetry, is the key to understanding vertigo.
It is everything. Vertigo is what happens when that symmetry is violently broken by disease or injury, creating a massive sensory conflict.
So let's say an infection knocks out my right inner ear. It just goes silent.
So your right ear stops sending its baseline signal. It's firing at 0 spikes per second. But your healthy left ear is still humming along at its normal 80 spikes per second.
To my brain, that looks like the exact same signal as turning my head, right? A difference of 80 spikes.
Your brain has no other way to interpret it. It misinterprets that pathological silence from the right ear as a constant, unrelenting head rotation toward the healthy ear.
So my brain thinks I'm spinning even though I'm sitting perfectly still?
Yes, and it's trying to correct for it. Your VOR fires up, trying to move your eyes to compensate for a rotation that isn't happening. That causes nystagmus, the beating eye movements. Your VSR fires up, trying to adjust your posture to keep you from falling over from a spin that isn't real.
And that massive conflict between what your vestibular system is screaming, we're spinning, and what your eyes and your body are feeling, no, we're still... That's the horrifying sensation of vertigo.
That's the sensory mismatch at its most extreme. The brain is trying to solve an unsolvable physics problem in real time.
What an incredible journey. I mean, we started out in this dense protective bone, and we've ended up mapping a signal from a single microscopic hair cell all the way to 4 different brainstem nuclei that are running these incredibly fast reflexes.
It all comes back to that simple principle: maintaining a continuous, symmetrical baseline discharge. Every piece of hardware, from the chemistry of the endolymph to the orientation of the kinecilium, is built to serve that one purpose.
The whole system is just this beautiful piece of comparative engineering, using fluid drag for one kind of motion and a heavy, dense, crystal-laden membrane for another.
It really is. It's a system built on subtle differences, on asymmetry being the compass, and it's running every second of your life.
It is. And, this brings us back to one of the most fascinating ambiguities in the whole system, which is a great final thought for our listeners to take away.
Absolutely. We said that the otolith organs, the saccule and utricle, use that heavy otoconal membrane to detect two different things: static head tilt, which is your relationship to gravity, and linear acceleration, which is inertia.
But physically, at the level of the hair cell, the shearing force they produce is identical, isn't it?
It is identical. The peripheral sensor itself cannot tell the difference between you tilting your head back while standing still and you accelerating forward very quickly in a perfectly level car.
Yeah.
Both actions create the same physical force on the macula. So the question we leave you with is this: if the primary sensor can't distinguish between these two very different real-world movements, how does your central nervous system so successfully disentangle them to know, without a doubt, whether you're just tilting your head or actually speeding up?